Hello. I'm going to give a brief introduction to the workflow before I introduce our speaker, Danny Miller, to share his experience with adaptive sampling. We will have an informal Q&A session, so please ask a lot of questions. And as this is an on-site only event, we're only going to take questions from the floor and not from the event app of the virtual platform. Also, Rosemary's presentation yesterday, when it comes to targeted sequencing, there's a number of options. You're all familiar with most of them. They all have advantage and disadvantage. Discussing those is not the remit of today. We're going to focus on adaptive sampling. Adaptive sampling is a very unique to nanopore method of targeted sequencing, with the key element being that you do the targeting on the device by a sample uploading of a bed file with the coordinate of the region you want to get. So I expect you're all familiar with our core chemistry, which is made of the pore, a motor enzyme, and the run condition that enables us to capture the flow of current that then we transform the signal into the sequence. And adaptive sampling is the process of ejecting the DNA strand in real time. It allows enrichment or depletion of the region of interest on the flow cell with no additional prep or specific time. It's really simple to use. You upload a reference file in Mino with the region you want to enrich or you want to deplete, and then you start your sequencing run. And then you have real-time base calling and alignment that takes place as the strand of DNA moves through the pore. If the strand is not part of the region that you're targeting, it's going to be ejected. If it corresponds to the region that you've selected, the sequencing will be ongoing. The Epitome desktop application offers really comprehensive analysis of the human genome in a single workflow. It's investigating human genomic variation comprehensively, including SNVs, SVs, and methylation, as well as copy number variation, short tandem repeat expansion, as well as providing annotation file and report for all those variants. Of course, it also gives you SNV's phase block. And the visualization of the map read can be done, and the different track can be done either within Epitome or on your favorite genome viewer. Before I give the floor to Danny, I wanted to share two examples. They're recent and they're really representative of what we see at the moment in the literature on how adaptive sampling is used in clinical research. So the first example comes from a publication by De Boer and their colleague earlier this year. The team is looking to establish a workflow for analyzing short tandem repeat in neurological disorder. They've created a panel sequencing for 9 genes associated with spinocerebellar ataxia and Fragile X syndrome. They're applying their raw data to reports, nanopore workflow on a sample from 12 affected individuals, and they achieve concordance with results from their previous diagnostic test in 22 of their 23 STRs. The take-home message from their paper is that the ability to concurrently detect SNVs, indel, and methylation really offers potential for one-test-fits-all solution in the future. The second example is by Di Letto and their colleague in Italy. They also published earlier this year. As part of the Teleton Undiagnosed program, that team used adaptive sampling, confident that it could get an answer. They targeted chromosome 5 and achieved about 100-fold coverage, specifically applying the Epitome workflow, human variation workflow, to look at variant in a child that had suspected Sotos syndrome, but for which exome and microarray were still negative. With adaptive sampling, they've identified 447 base pair insertion within exon 13 of the gene NDS. And then they did RNA analysis that suggested defective splicing. So these two examples really show you how adaptive sampling is simple, yet it's very powerful for answering questions that couldn't really be answered by other technology. So now I'm delighted to introduce our guest, Danny Miller. Danny is assistant professor at the Department of Pediatric, Division of Genetic Medicine, and the Department of Laboratory Medicine and Pathology at the University of Washington. He is also an attending physician at the Seattle Children's Hospital. His laboratory is developing long-read sequencing-based clinical genetic tests with the goal of increasing the rate of genetic diagnosis, reducing the time required to make genetic diagnosis, and lowering barriers to obtain comprehensive clinical testing. Over to you, Danny.
Thank you, Cora. I appreciate that. I appreciate the introduction. When I was asked to do this, I actually wasn't sure what I would talk about initially, because now that I've become a PI, I'm actually not allowed in the lab anymore. So doing the workflows now, I don't really do. So what I wanted to do is go back to some cases that we've solved using adaptive sampling, and just take kind of a historical how it's changed over time, and kind of things we ran into along the way that are still relevant today. This is the required disclaimer, and then conflicts. So the first case I want to show is actually one of the very first cases we solved using adaptive sampling. We sequenced this probably back in 2019, 2020. This is back when it was known as read fish or read and tell, so before the name was changed. This was a kiddo that we saw in clinic who had intellectual disability. He had multiple congenital anomalies. We did a karyotype, we did an array, and you can see on this karyotype that he has multiple translocations or rearrangements among these 4 chromosomes. And the question we had at the time was, how do we actually resolve these? So if you were in the last session, you saw Sean show that assembly. I forget which chromosomes it was, but between, I think, 2 and 14, where there were multiple rearrangements. And we wanted to know how we could actually resolve this one using adaptive sampling, or targeted long-read sequencing is also what we called it. Just to kind of get to the chase, we targeted about 120, I think, megabases of space across those 4 chromosomes, and we were able to actually reconstruct or rebuild copy number plots using adaptive sampling output. So you can see that there's a loss, what's labeled 4B, and then there's a couple other losses there as well, 4D and 4C. Some of these showed up on the array, some of them did not. What was more interesting about this case is that we were able to use the adaptive sampling data to completely reconstruct the structure of those 4 chromosomes. So we knew the exact path that the breaks took across those 4 different chromosomes. We did have to go through and do this manually and then draw this manually, but the point being, we used a single MinION flow cell using blood-derived DNA to actually identify all the copy number changes and then the rearrangements that were involved in these 4 chromosomes. The result of this clinically, there was a change. He had a breakpoint that bisected a gene associated with hypertrophic cardiomyopathy, and he now gets echoes every couple of years just to make sure that he does not develop that. The second case is one that we used to kind of prove that we could use both adaptive sampling and perform methylation analysis at the same time. These are 2 kiddos who have a condition called Baratella-Scott syndrome. We saw them up in Seattle. And the story was, before they came to clinic, they had an array. We knew they had a deletion around the gene associated with the condition, XYLT1. But we didn't know what the second hit was. This was a recessive condition. Over time, an investigator who was at UW at the time, who's now at St. Jude, Heather Mefford, she figured out that they actually had a repeat expansion in the 5' UTR of the gene. But to solve that, she had to do whole genome sequencing, she had to do bisulfite sequencing, repeat prime PCR, she had to run a Southern, all these things. Our question was, could we use long-read sequencing, and specifically adaptive sampling, to get the same result in a single test? And the long story short is that we did. So again, we used blood-derived DNA in this case. And actually, now that I see this, I think the dad was saliva-derived DNA. But you can see on that plot in the proband, we see an expansion of the target region. In mom, we see a separation of the premutation allele and the wild type allele. And then in dad, we get very few very few reads. This was one of my earlier runs, which is why we didn't have such great data. Also, the methylation calls were done using nanopolish, which didn't always convert the reads. So that's why some of those gray bars are still gray, and they're not red, which means methylated, or blue, which is unmethylated. The next example is an example from the complex GNAS locus. This is associated with a couple of conditions, one of which being PHP1B or pseudohypoparathyroidism type 1B. The idea here is basically that it's an imprinted region, and depending on whether or not you inherit the region, a pathogenic variant in the region from your mom or your dad, you can have one of the conditions that are associated with the gene in this region. So this example is just to show that using adaptive sampling, we're able to target the region. We're able to identify a transposable element, which is shown in panel A up there, which was the pathogenic variant for this individual. And then if you look down in panel B, you'll see that there's a block around a region which is blue, meaning it's hypomethylated. But then on the bottom, you'll notice that there's some red. So actually, the bottom one is the wild type. So you see hemimethylation on part of that region and then hypomethylation on the other. But the one above it, the 208.3-1, that's the individual who inherited the pathogenic variant. And in this case, she lost methylation of that region, which gives her that diagnosis of PHP1B. This, again, was blood-derived DNA. This was sequenced using adaptive sampling on a Promethean. And we get really good results when we use adaptive sampling on the Promethean. We actually don't even shear the DNA. We just put in whatever we get, and it works really well. So that's all. Those are the three examples I wanted to show you. I've, of course, thanked the lab for the work they do on this. And then I think we're happy to have a conversation or take questions.
So do we have any question from the audience? I forgot some question. You've been using both adaptive sampling and whole genome. I mean, is there any particular application or reason that you pick one over the other? Because that's something that we get quite a lot.
I can talk about it, a reason that we might use adaptive sampling today. I will say that if I have an unsolved case, I actually prefer to do whole genome sequencing, just because I don't always trust that we know where to look, or, we know what to target with adaptive sampling. But there are cases where we do use it, and one example is if we get a complex structural rearrangement and we want to recover longer reads covering that rearrangement, we'll do adaptive sampling, again on the Promethean flow cell, and we recover reads hundreds of kbs long by doing that, which lets us resolve that complex structural variant.
That's something that comes a lot. And I think the second most common question I get is how difficult it is to design a good bed file to do adaptive sampling and how flexible that is.
Yeah, we have a script that does it, but I do always wonder, why doesn't Nanopore provide a tool to do that? Maybe they could.
There's quite a few, actually. Which one do we pick to support? Alden, thank you.
Hello, thank you for the talk. Can I ask, do you get even gene coverage amongst all your gene targets when you do adaptive sampling?
For the ones we target, yes.
So it's even?
Yeah, unless there's a aneuploidy copy number variant, something like that, you're going to see differences. But for the most part, we get really nice coverage. Yeah.
Okay. And what's the coverage about?
I mean, it depends how long you sequence tell, and it depends if you shear your DNA. I would say, and I think Joy's over there somewhere, if you shear to about 20 kb or so on a Promethean and you sequence for 24 hours, you'll recover about between 40 and 60x of those target regions. It's also a function of how much space are you covering, right? I think the recommendation from Nanopore is 2% of the genome. We do tend to go a bit higher in some cases. 4 or 5% seems to work well in our hands. So we kind of play with those numbers just to hit the coverage that we want.
Thank you.
Mina, thank you.
Thank you very much. Great talk. I have a question regarding the output. Is there any observable differences in yields you can get from adaptive sampling flow cells versus whole genome sequencing flow cells?
Yeah, I mean, you could get 3 or 4 times as much, 5 times as much coverage using adaptive sampling. I think I heard the question. Sorry, it was a bit...
Just the total amount of yield. How much gigabases? Is there a difference between adaptive sampling or versus whole genome sequencing? Just in total gigabases output per flow cell? Yeah,
I'm going to ballpark and say you get about half when you're just targeting certain regions. So we might get 100, 120 gigs doing whole genome, and we'll get 50 or 60 gigs doing adaptive sampling.
Thank you.
One thing that I love about adaptive sampling is analyzing the off-target regions. So you still sequence about 500 base pairs of the regions that you reject. So you get to do genome-wide copy number analysis outside of the genes that you're targeting, which I think is a really cool, just kind of like for free application of it. And then the other thing is, of course, you can look at methylation in those regions. So Matt Luce's paper that just came out recently, the brain tumor one, I mean, you're getting the methylation calls off of those off-target regions, which is a really nice use of the data.
And for people who came to the breakout this morning, do you have any visualization tool at the moment, or are you guys working on things that will enable to have some reporting specifically on the target region and some level of copy number reporting?
Yes, yes to both. Yeah, for copy numbers for sure, and then methylation as well. Yeah.
Yeah.
Just curious, do you do washing and reloading as well with this?
We do, yeah. We get you watch your pores. So on a Promethean flow cell, if they get to about 2,000, we'll do a wash and a reload.
Right.
It also depends on the application. Joy just gave a talk about phasing for ASO design.
Yes.
For that application, we typically get enough after 24 hours of sequencing. But if we're trying to resolve a really complex structural variant, we'll tend to run it for 3 days and get as much data as possible, because we just don't know if we're going to get reads long enough spanning that complex variant.
Okay, thank you.
Yeah.
More question?
Someone in the back.
Question. So this is just a general question. Does it matter what size of reference you're using for your adaptive sampling in terms of the output of your data?
I'm probably not the best person to ask, because I tend to ignore the recommendations and just do what I think is going to work. So for the human genome, I think the recommendation is to target about, again, 2%, but somebody should check me on that.
But between 1 and 10.
1 and
10%.
1 in 10?
Yeah.
Oh, I think it's not 10. It's lower than that.
Yeah.
We'll target up to like 5%, and we get really nice recovery. It is kind of crazy, though. We'll do a human genome. We might target 5% of the genome, and then you look at the rejected reads, and you get a 30x genome in the background from those rejected reads when you target that much. And you show that to people, and like, wait, what? It's really cool to see.
If you have a region of interest that you want to target, do you also want to keep some flanking regions just in case the read started? And then the question is how big should those flanking regions be?
Yeah, that's a great question. It kind of depends a bit on your average fragment length going in. So imagine that you have fragments that are 100 kb. You want to make sure you're grabbing the fragments that start 50 kb out that go into the region, right? So you definitely want to have buffer. It depends a bit on fragment length. If you shear, we tend to do 50 or 100 kb on either side. One thing we don't do that we should do is be strand specific, because you don't really care if a read starts outside of the region if it's going away from the region. Right now we're not strand specific, but I do think that's the better way to do that.
We've got a question there from Belen.
Hello, here. Do you know, or do you have experience of any genomic regions that are less efficient in adaptive sampling because of the nature of the region? Do you have experience on that?
That's a good question. I've never done something, well, I think a long time ago I did something really dumb, which was target transposons. I targeted lines and alus or something, and I don't think I got much out of that, but that might work better now. I've never done something, but I've wanted to, trying to target heterochromatic regions, right? Just ignore all euchromatic sequence and see what you get if you just target, centromeric sequence, things like that. But I don't know how well that would work. So that's not a direct answer to your question. It's just kind of like I don't know, but here's some things that could be interesting.
Okay. Do we have one last question? It was interesting. I was just in a meeting now where people were saying they see some specific enrichment sometimes on some regions. So similar, the opposite question from Belen. If you think there's regions that are particularly easy to target for adaptive sampling?
No, I don't think there are regions that are I mean, it's just DNA to the pore,
Okay.
right?
Yeah.
So the pore doesn't care, right? Yeah. So.
Yeah, cool. I think we are run out of time. So thank you very much all for joining for this demo. Special thank you for Danny again.
Yeah.
You've got another presentation in like 2 minutes, so we're not going to keep you. We're, sorry, I'm getting lost. Thank you again, and we'll see you again at 3:50 back to the auditorium for the closing session, which is the panel discussion on clinical genetics. Thank you, Jill.
Thank you.
You very much.